immunological diseases. So maybe it's best answered by you.
Well, one of the areas. So I think that indeed, nowadays there's a trend to make every cancer almost individualized, and I think that that is where one of the key opportunities lies. So if you again envision a patient journey from diagnosis, cancer is diagnosed. And also sequenced with its individual epitopes, your personal collection of immunogenic epitopes where you could be vaccinated against. That is translated into a messenger, your personal messenger RNA cocktail on site in your messenger RNA printer, and then formulated on site with a lipid nanoparticle in a similar fashion as the COVID-19 vaccines to try and stimulate your immune system to recognize the cancer cells. I think that that is already, I think, being developed by BioNTech, for example, and CureVac, to try and build this pipeline of activities where individual diagnosis, individual cocktail of messenger RNAs is translated into an individualized product. And I think that would be best served by doing it all on site.
Yeah. Last couple of questions, and maybe a few quick ones I can throw out there. This is from Hanine Ali Bouchetti from the University of Birmingham. Do you envision NanoSPRESSO being adapted for other drug delivery systems, such as polymer-based nanoparticles?
I think we can do. The moment that there's a self-assembling system that requires the encapsulation of a compound and is driven by the physical interaction, essentially, of your polymer and the drug, I think there's nothing against it in combining it. I mean, we've made polymeric micelles in the past, which is essentially the same process. There's the how these compounds should work. A messenger RNA should translate into a protein. So your lead compound and the identification is very fast, and your mechanism of action testing is complete in weeks. And importantly, and this is really crucial when you want to develop a pharmaceutical formulation, essentially it's always the same molecule. It only differs in length and sequence. And that is something that is shown here. So on the right-hand side, you see proteins, but it also applies to conventional therapeutics. Whenever you start on a project, you work with your therapeutically active molecule, but you never know what type of characteristics it will possess. Will it be positively charged, neutrally charged, negatively charged? Will it be very sensitive to light, to humid conditions? You never know. So every project starts essentially from scratch. If you take the opposite, on the left-hand side, the nucleic acids, it's always essentially the same molecule. It only differs in length and sequence. By changing the letters in the alphabet, you change, for example, the messenger RNA and change the protein that will come out of it. But essentially, it is the same molecule. So the lessons that you learn while developing your formulation for the first time, they can be applied to the subsequent formulations. And that is really an asset in drug development. It can really speed up things. So how to overcome these difficulties? It is a wonderful molecule, but it is difficult to deliver. And there goes a big shout out to Professor Peter Cullis, University of British Columbia. He was one of the first to envision how this could be done and how this could be done with lipid nanoparticles. So with lipids, you can actually encapsulate the therapeutic nucleic acid into a nanoparticle. And that can be done by self-assembly, because these lipids will organize themselves in an aqueous environment in such a way that they encapsulate the nucleic acids into nanoparticles of approximately 100 nanometers. Of course, this needs to be very carefully controlled. Otherwise, you get particles of all different sizes and they go all different places. But if you mastered the conditions under which these particles are manufactured, you get very reproducible, very nice nanoparticles. And these can help you deliver your therapeutic nucleic acid to the right site. So this is an advanced therapeutic which almost does everything you could want for. So it's intravenously injected. Subsequently, it arrives at the surface of the target cell. It is then internalized. The therapeutic nucleic acid is allowed to escape by the help of the lipid nanoparticle, and then the therapeutic nucleic acid can do its job, in this case, nuclear translocation, integration, and payload expression. But essentially, all of the therapeutic nucleic acids, once they have entered the cytoplasm, they could do their therapeutic activity. And the first lipid nanoparticle filled with a therapeutic nucleic acid came on the market in 2018. That was Onpattro. Not a moment too soon, you could argue, because that allowed us to build upon that knowledge that was generated during the production of that particular drug molecule during the COVID pandemic, of course. And that is not a moment too soon, because within a year we were confronted with a pandemic that required us to really learn from all the lessons that we've learned on the left-hand side from patisiran. So this drug, patisiran, provided a recipe of how to make lipid nanoparticles loaded with a therapeutic nucleic acid. You can see a variety of different payloads. So you see the siRNA for a particular type of amyloidosis on the left-hand side in patisiran. Then you have the two COVID vaccines in the middle and a new CRISPR-Cas-based advanced formulation that is currently in clinical trials on the right-hand side. And essentially, they are all built on that same recipe of 4 lipids together with the nucleic acid, form a nanoparticle and can be used therapeutically. If you look at the COVID vaccines, of course, everybody was focused on scale-up. During COVID, new factories emerged where lipid nanoparticles were churned out by the kilogram. At the same time, in our lab, we make lipid nanoparticles, and I think I can say we can make nanoparticles at a very high quality. I would gladly inject myself with these lipid nanoparticles because I think they match the quality of industrially produced lipid nanoparticles. And that can be done by the virtue of microfluidic mixing. You can get very reproducible mixing if you do this on a microscopic scale. So the self-assembly can occur in these types of microfluidic chips. Here you see the 2 fluid streams, one yellow, one red, resulting in an orange mixture. And that is essentially what needs to be done. The nucleic acid needs to be encapsulated in the lipid nanoparticle under reproducible conditions. And within such a microfluidic chip, you can essentially always replicate the same conditions, provided that you keep the flow rate and the flow rate ratio constant. And that's, well, the fact that we produced in our lab high-quality nanomedicines, which I gladly inject into myself, was basically the foundation for NANOSPRESSO. Not in the least because I worked together with Huub Schellekens at one point, who devised or thought about the Biospresso. That was perhaps a bit too early for its time. He envisioned a individualized protein therapeutic production facility on your kitchen top, where bacteria would be engineered to produce the antibody of choice that would help you cure your therapy. That is still difficult, I think, but the thought lingered in my mind. And I think with nucleic acids, all of a sudden, the concept can become a reality, because combining nucleic acids and lipids in a blister and do microfluidic mixing inside a cartridge should allow you to give always reproducible lipid nanoparticles. Combine that with a cartridge inside an activation station, and that activation station should take away all the burden from the hospital pharmacist regarding the quality control. So provided that the pressure buildup in your cartridge and all the loading controls and volume dispenses has happened correctly, the green light should appear, and as a result, the hospital pharmacist should sign off on the quality, because all of the quality controls is embedded within the activation station and doesn't require a subsequent testing of the final product. And then you arrive at a vision like this, where on the left-hand side you obviously have your Nespresso machine with all the tastes and that are available, and on the right-hand side you have your activation status with the different cartridges filled with the nucleic acid of choice that is therapeutically active for an individual patient. You load the cartridge, put them in the activation station, and as a result, it will produce the high-quality lipid nanoparticles within your hospital pharmacy. There are new developments going on. This is an example of CureVac's messenger RNA printer, as they call it, where in a completely automated fashion, even locally within a hospital, you could produce GMP-grade messenger RNA. And also companies like Abogen have a completely automated suite in which they can make messenger RNAs on demand. So you also see there that from a production perspective of the active ingredient, that even is scaling down into local hospital settings. So with that, on the left-hand side, we have our ingredients supplied with a variety of companies that participate within this consortium. And within the middle, we have our academic institutes that participate to try and make NanoSPRESSO a reality, where we have the machine, the characterization and performance, and of course, all the legal regulation and societal challenges that surround it. And finally, of course, we all do this to benefit the patients.
Entails the conversion of a substance A into a substance B. And when this does not happen in the right way, then substance A will not be converted into substance B, and it can lead to accumulation of a toxic compound and thereby cause symptoms, or it can lead to a deficiency of substance B, a compound that might be needed for the functioning of the organism. And depending on the enzyme or the specific mutation, essentially any organ can be affected and any symptom can be a presenting symptom occurring at any age. In the Netherlands, because that's where I work, we know that approximately 10,000 families are affected by one child or more children with a metabolic disease. And with the integration of more and more genetic screening, this number is expanding gradually. Many of these diseases are untreatable, but for some diseases we do have treatments. And we've focused with our treatments historically on the metabolites that are altered. So if one of the substances becomes toxic, then we try to avoid production of the substance by giving patients, treating them with a diet. On the other hand, the problems may also arise because of the deficiency of a certain substance, and we've been used to treat that by providing that substance. But this can only treat a subset of patients. With evolving possibilities, it became possible to also directly target the enzyme that dysfunctions, and we can do that either by providing a cofactor for the enzyme, or we can deliver the enzyme in a pure form by itself, or through an organ that produces the enzyme, which in the case of metabolic diseases is often the liver, because the liver is a very important metabolic organ. But it might also, for example, be hematopoietic stem cells, because they can also cross the blood-brain barrier. But even with these therapies, still many diseases stay without a good treatment. And I've been thinking, why are we so poor or so bad still at treating these diseases? And I think it is because metabolic diseases represent such a large group of so many different individually very rare diseases. And we've always been focused to try and really understand one of those diseases and try and develop a treatment for that. But with the increasing number of diseases, which are now over, well, for metabolic disease, we know now that there are over 1,900 diseases, but for rare orphan diseases, even over 8,000 diseases. So I think it is now time to try and look for a more general approach, which we can then target to the individual rare disease. And when I think about what is general among all these diseases, is that they are all genetic diseases that result in an enzyme deficiency. So when focusing on that genetic cause of disease, I will first focus on the affected mRNA that Ray also talked about already. So mRNA can be used now as a therapeutic, and it has been used in the COVID-19 vaccine, of course. And Ray already mentioned Peter Cullis as a key person in developing mRNA medicines, but Katalin Kariko is certainly also a key person in trying to make these mRNAs usable for medical application. Once you use this technology, the mRNA encapsulated in lipid nanoparticles, but inject them intravenously, then almost all the lipid nanoparticles accumulate in the liver. And these are experiments that were actually done in Ray's lab, where you can see that we used a reporter mouse in which the mouse was administered with an mRNA that turns the cells red. And here you can see the organs in which most of the cells turn red, and you can see that with increasing dosages, almost all liver cells are reached with this lipid nanoparticle. So we thought that is really interesting for metabolic diseases, because the liver is such an important organ for metabolic diseases. Maybe we can give the deficient mRNA or the mRNA of the deficient enzyme to treat patients with metabolic diseases. Unfortunately, Moderna thought the same, and they developed a treatment, a first-in-human trial for two different metabolic diseases, propionic acidemia and methylmalonic acidemia. And these are two severe metabolic diseases in which the liver cannot metabolize certain amino acids, and that results in the production of toxic compounds that can affect different organs, like, for example, the brain and the kidneys, but also the heart and also the optical nerve, so patients can become blind. So it's a severe disease for which currently we don't have a good enough treatment. The treatment that we are testing right now is the mRNA treatment for MMA. That's what I'm talking about, but we also host the PA trial. But for MMA, we encapsulate the encoding mRNA that encodes the deficient enzyme, which is the MUT enzyme in this case. It's injected intravenously in the liver. The formulation will arrive and the mRNA will enter the cells, where it can be translated into the MUT enzyme, which can then be translocated in the normal physiological way to the mitochondria, where it will dimerize into the homodimer, which can then perform its activity. This is an international study, which is a dose escalation study. And here you can see the first results of the first 3 cohorts, which were the lower dosing cohorts. And we are now in cohort 6, in which patients receive higher doses of 1.2 milligrams per kilogram already. But what you can see is that we here look at the toxic products that are produced in the patients. It's methylmalonic acid and 2-methyl citrate. And you can see that in the first cohort, where we used a very low dose, there was no effect. But with increasing dosages, you can see that there is an increasing effect in reduction of the production of the toxic compounds. And with the higher cohorts, we even see this stronger. We have treated 3 patients in the Netherlands now. This is one of the patients. He started in cohort 3, and he started in cohort 4 actually. And he was a very shy boy with not very much energy. And upon treatment with this treatment, we can see that he became much more energetic, and the parents really say it's a totally different child. And actually, we see remarkable clinical effects in all the patients that have been treated so far. For the propionic acidemia trial, the interim analyses have already been published, and there they also see beneficial effects of the treatment. And by now, there are mRNA trials that are either running or being prepared for a number of other metabolic diseases, which primarily affect the liver. The major limitation, however, is that the mRNA expression is only transient. So it is only expressed for a couple of days, and then the enzyme is expressed for a couple of days, but then you see that the effects are fading. And actually, in our older patients, they really report that they feel well after the treatment, but after a week, they start already feeling symptoms again. And that means that these patients really need to come to the hospital every 2 weeks to receive an intravenous injection for their medication. So I think this is a huge development for metabolic diseases, but we're not there yet. And I think we can now focus really on the disease-causing mutation, and I think that is what we should do. Because this was enabled by the CRISPR-Cas9 technology, which was first described by Jennifer Doudna and Emmanuel Charpentier, who won the Nobel Prize in Chemistry in 2020. And they showed that the CRISPR-Cas system can be used as a very precise molecular scissor that can cut the DNA at an exact location wherever you want to cut it, basically. This is really exciting, and you can really cut the DNA at a place. You can use it to repair a genetic mutation, but that is quite an inefficient process, and rather the cell will react on the double strand cut in the DNA by trying to repair it directly, which is a sloppy procedure and which leads to the integration of insertions, or can insert deletions in the DNA, which will rather silence a gene than correct it. So that is why David Liu thought of using a slightly less active Cas9 enzyme, a nicking Cas9, and if you fuse that to another enzyme, for example, a deaminase enzyme, you can deaminase certain bases and thereby convert, for example, an A into a G or a C into a T nucleoside. So this is very specific and really can help to correct a mutation, but it's only applicable to very specific mutations. So then David Liu thought of another system, and he thought of combining and fusing the nicking Cas9 enzyme to a reverse transcriptase enzyme, which is basically a writing enzyme that can rewrite parts of the DNA up to 20 nucleotides, but by now even longer stretches. And thereby you can essentially edit any mutation that you want to edit. So what if we would exploit this transient expression for direct in vivo gene editing? So giving the modified RNA that encodes the gene editing tools, encapsulate that in lipid nanoparticles, inject that intravenously, and then it will arrive in the liver where it will be expressed. It can then correct the mutation. And because the expression is only transient, the tools will be cleared and you will have a permanent cure of the cells with this transient treatment. So we think this is really a game changer. And I think that because for a lot of metabolic diseases, we know that we only need to correct approximately 5% of liver cells to resolve most of the symptoms of the patients. And in the first animal studies that were done using these lipid nanoparticles, we saw that for base editing for a certain disease, we could reach much higher levels in liver cells of mice and also in non-human primates. And we also saw that it was safe. And by now, the first human trials are ongoing for 2 different liver-dominant diseases, and in both beneficial results have been reported already, but these are, of course, preliminary yet. To target other organs, another system has been developed in which you can encapsulate the ribonucleoproteins in virus-like particles. And the very first animal studies for that have also shown quite promising results, I'd say, in mice, where they've seen that over 60% of liver cells were edited after 1 intravenous injection, and also retinal cells and even brain cells could be targeted after localized injections. So we tried to use this to develop a gene correction therapy program where we have developed a specific reporter that we can use to tailor our gene editing tools to the patient-specific mutation and the mutational context. We have a large biobank with organoids from a lot of patients and from different organs that we then use to target and further refine our treatment to the organ-specific and patient-specific context. And then we aim to develop these into personalized therapies using the NANOSPRESSO approach. And we first focused mainly on the liver because it's the easiest organ to target, but with some recent financial support, we are now also focusing on other relatively easy to target organs like the bone marrow cells, the eye, and not easy to target, but very, very wanted for metabolic diseases, the brain. And this may sound like science fiction, but just a month ago, it has been shown that this is really possible. This is in the United States, where they've treated a very first patient who was diagnosed after birth with a rare genetic metabolic disease. And within 6 months, the treating team, in a collaboration with regulatory authorities, but also with industrial partners, they were able to transform this lethal diagnosis into a treatable condition. And they've treated this patient now with 3 doses, I think, of base editing treatment to try and correct the underlying genetic disease. Of course, this was the ideal candidate, the ideal patient for this situation. This was a patient with a liver-dominant metabolic disease in which we know that the liver is the easiest organ to target. And this is a disease where you don't need to target all the cells to resolve the symptoms. Parents were open to this. This was a base editable mutation. That's also very important because base editing is still more efficient and more precise than prime editing. So I think if we want to translate this treatment strategy to the many different patients with metabolic, but also other genetic diseases, we will need to also to try and target also other organs, but also involve prime editing, because with prime editing, you can address essentially any mutation. And also for prime editing, there are encouraging developments to try and develop these also into clinical therapies. So with that, I think it is prime time for patients with metabolic diseases, because in addition to the treatments that I explained already before, we now have new possibilities using mRNA therapies, but also the precise gene correction therapies, in addition to the gene therapies, the gene augmentation therapies that I didn't mention today. It's not only us, the researchers and the medical doctors that are excited about this, but also the patients themselves who try to gather money, to collect money, to invest in these developments.
The need to kind of form a basis or a categorization system whereby we can refer to this definition to kind of lower that threshold for approval and kind of build on existing knowledge, leverage that knowledge for new products. So with this in mind, I kind of developed this, categorization system, which is what regulators are already kind of doing. So they are already making these considerations, but I've just put it into a decision tree, essentially. So we're still going to be considering the chemical synthesis or the biological production, as that does have an impact in terms of the regulation. But in terms of for RNAs, they do have characteristics that have regulatory implications. So you can have binding, sequence-specific hybridization, or insertion of an entirely new sequence. And these are the 3 kind of main broad categories. And then you can have subdivisions for the common chemistries that do impact the safety profile. And the benefit of this is that it will enable the pooling of data and then permit us to use prior knowledge, which is essential against rare diseases with small patient populations, where you are restricted in your ability to gather kind of mass scale clinical trial data. And especially we're talking about something like NanoSPRESSO, this kind of thing matters because we envision it as a platform technology. To very, very briefly discuss lipid nanoparticles, it's only 2 slides, I promise. So in this device in NanoSPRESSO, the lipid nanoparticle is not the active substance, and that is within the European regulatory system overall. In the COVID vaccines, it's considered an excipient, not an active substance. It is always just a carrier, an excipient. The nucleic acid that it carries is considered to be the active pharmaceutical ingredient. So how do regulators look at the lipid nanoparticle component? Well, the reason I'm interested in looking into this is because the lipid nanoparticle does have immunogenicity effects, and we know this especially after COVID, but there is no definitive framework for regulating them. So I really wanted to kind of look into this and see if there's any characteristics that do impact the safety profile for lipid nanoparticle containing products. And this will really help us to advise the developers, in our case, NanoSPRESSO, for what kind of evidence they might need in terms of controlling the safety aspects of a lipid nanoparticle in a platform technology. And importantly, the findings that we're going to gather from all of these areas should have impact for the broader community and not just for NanoSPRESSO.